Thank you for introduction. Really, I studied the cultural network from 2010 years, I think. It's about 15 years. Mechanism of plasticity of the neural network. First of all, I think I want to give some definition. What definition? What? No? Okay. When we say about the cells, today we say about the cells. We need to say that in any levels of the organism, there are cells. On the all organism, on the organ, on the tissue, on the cells, and between cells there is synapses, and synapses include a lot of molecules. And when we want to take from this chem only cells, we need to understand that it is very, very artificial conditions. And I read a lot of text when I find some definitions. Some of the scientists which study network use a neural network. What does mean neural network? From point of view, from my point of view, neural network refers the whole network which is responsible for the decision of the big higher order task. But some of the scientists use a neural circuit. What is neural circuit? It's not the same like the neural network, because as usually if you use patch clamp, if you use measurement of the LTP, LTD, and STD spike time dependence plasticity, you usually use neural circuit, which include only some of the cells, 2 cells, 3, 5, 7. But neural network include a lot of cells. And in my work, I use term neural network when there is a lot of cells. I understand that there is no show, yes? No? Ah, okay. And from the point of neural networks of the brain, it is the same, the set of synaptically connected neurons involved in the receiving, transmission, storing, and reduction information. And if you compare neural network with the neural network artificial program, we need to say that we may choose some general properties of the all networks, biological networks. Now, first of all, is these properties include the properties by which regulated of the properties of the membrane. If you change the membrane excitability, we change the properties of the neural network. Neural functions can change and adapt to the different environment. And today I show some examples of this changing according to the environment. The neural circuits are genetically programmed, and maybe these properties permit to use cultured neurons in the plate and grows a lot of time and look for a lot of the same properties like in vivo. The next, in the process of brain development, the local change in neural network are possible with the formation of new connection between neurons. We call these properties like plasticity of a neural network. Neural networks interact with the other cells, and today we say about that because around those neurons there are many cells like the astrocytes, microglia, and now we cannot consider separately neural network, but still now we say about the properties like the properties of the neural network. But when we say about that, it means that we consider interaction between these cells and between the astrocyte cells, microglia cells, which help to work of this neural network and may modulate its plasticity. Resolution of neural networks. Neurons work in the distance of micrometer and temporary millisecond. And the neural networks may form functional system, I think, because anytime we may investigate this neural network from the point of structure, structure may be changed, but very rarely. Functionally systems, anytime be combined and direct on the some task. So, mm, mm. Why we take cell culture? Even there are a lot of mouse or rabbits, monkey, and so. Cell culture now is very useful tool for the cellular and molecular biology because we may see the single cell, we may do PET clamp, we may visualize, we may apply a lot of substances on this cell, and cell culture provides scientists and researchers with excellent model system for studying not only the normal physiology and biochemistry of the cells, but a lot of pathological process without living organism. And really, it's less expensive, but what I'm not sure with this topic because I work a lot of time with the culture, and I know that it's very expensive, but from the point of some people consider that less expensive, but more humane to grow cells than mice, rabbits, even more so dogs and primates. So we may get on the preclinical study some very important materials without use very expensive and animals. And now from the point of the modern preclinical search, to use cell culture must be like the law. And when we say about the culture, cell culture, we use primary cell culture. What does mean primary cell culture? It means that you take the tissue, dissociate these cells, all cells, and calculate, plate in the well all these cells. What cells? The first of all is neuron cells, the yellow color, plus astrocytes. Astrocytes, you saw today this picture, connect with vessels and synapse, but never time connect with the neuron. And if we consider neuron network, like a lot of neurons in one set, the astrocyte site connect to each other and form special astrocytes network, but the size of this network significantly less. And it's usually rise near the active neuron synapses. Plus, microglia may decide be synapse in the network or no. Microglia not defense only factor, but is very modulator factor now. And a lot of material, a lot of study in modern neurobiology concerned the function in the neuroplasticity of the microglia. Oligodendrocytes and what yet? Extracellular space, extracellular matrix. Our team many years studied the rule, the function of extracellular matrix, and we consider from the point of astrocyte membrane to neuron membrane and molecular network of extracellular matrix like the tripartite synapse. And from the modern approach, we use the technology brain on a chip. What does mean brain on a chip? Brain on a chip, it means that we take a special microfluid chip, not maybe with a microelectrode array, maybe without, and combine a lot of tissues which are dissociated from the different part of the brain and apply and connect with each other through the special channel and consider the different part of the brain like the whole brain, but on the tip. Many years ago, we cannot think about this technology because there are no microfluid technique. And we use the general plate when we drop some amount of dissociated cells on adhesion material and observe their growth, the development. Maximum we growth this culture in our laboratory is about 8 months. To that, this tissue is really was tissue swim in the from the electrodes and not distracted, but we cannot measure electrical activity because there was a lot of astrocytes, fibrosis. So brain on chip system interesting not only for scientists when investigate the drug development for the preclinical search, but using this system, we may investigate the mechanism of the functioning of the central nervous system in normal and pathological conditions. We may change any condition, pH, temperature, oxygen, some toxical substances, and extracellular matrix, and so. And we may find some very interesting dependence for neuromorphic intelligence to study the mechanism of the information processing. We consider this network, neural networks, on the level of the cells. But today we saw in the presentation of Olga that we may see in the brain using the miniscope and see really a lot of cells on the cells precision in vivo. It's very useful as really, but my miniscope do some trauma is really, and we need to understand like the very useful tool like the slice miniscope, the same damage some tissue. But here we grow the cell culture, for example, from hippocampus, from the embryonic cells of the mice. We take in the last day of embryonic, on the 15th day, on the 15th days, there is a stop program of the stem cells and all neurons are differentiated. And as a result, there are a lot of connections between the cells. When we dissociate, we didn't damage the connections between the cells. We play it in the special conditions and grow this network, like the whole network, like the without any damage. And really, The more interesting questions, I think, from you must be how you talk with this culture. There are no inputs, there are no outputs. Not see our culture have no to see, to talk, to move, and so. And there is next specific term today. Usually, all of neurobiologists know burst. Burst, it is a series of repeated spiking of the neuron. But here we use this term like the network burst. What does mean network burst? Network burst, it is a special set of the spiking sequence which we see on all electrodes in some time. Our time, scientists artificially, I think, from the experience here, take the time is about 3 milliseconds, what as can you see here. When the distance between two single spikes cannot more than 100 milliseconds. And if the less distance duration between the first signal and the next, we consider this a set of spikes like the network burst. On the screen of oscillator, you see the picture side. All these spikes, like the black point on this picture, you see that during the culture, despite its will be is about 8 months, culture work using the constantly repeated rhythmical activity, maybe more simplest, maybe more largest, more complex, but despite that, it is the same rhythmical activity, very similar like in the sleeping state or in the initial days after the burning, very early postnatal period. And then you see on under the it is electrodes. It is a time when under the some electrode there is rising the spike. As then you see with this electrode, it was the first electrode, maybe number 50, for example. It is number 60, it is more, and so. It means that from the point of mathematical analysis, mathematician analysis, more easy to count the initial pattern of the spiking activity. And using this time, this delay, we may measure how this excitation through the network be spread. We call this spiking pattern like the spiking signal. And with cell culture from the different tissue, we may measure this spiking signature, and as usually, it's really like signature, will responsible for the structure of the sum of the cell. There are neonates, there are laborant or young scientists, we take the drop of the suspension of the cult and drop. Drop. One of them, what I know in my team, there are 3 specialized in the very culturing. Can be do very rarely and so. And depends on the task. If you want to investigate lots of time, some propagation of the signal, and we want to stimulate, and if you want to learn this culture, we use students, we do dance culture. If you want to do cytochemistry, or we may mark neurons, we take orally culturing neurons. But its influence on the performance of this signature and on the performance of this spiking build. But it's one of the disadvantages of the culturing. You need to have automatically very equal plating for the studying the same dependence between some factors which you investigate and this culturing. The next measurement of the activity of this network, we may use message of the graph, and as a result of the detection of the we may measure the cells, but not cells. Cells is not cells, it's electrode. Electrode, which measure electrical activity from the surrounding cells, may get a lot of inputs, or may have no any inputs, or maybe only one input. This electrode, which, on which located with the cells on electrode, may send information to one cell, maybe two more cells, and so. The infrastructure of this network may have some hubs. And during the development, this structure with these hubs be changed, but we know this dependence. And we know that if we have no any change in the surrounding, and we maintain the homeostasis, there is no changing of the structure. But if only some of cells be died, or some of cells will feel more bad, for example, there is hypoxia, ischemia, or some toxic substances, the structure be changed. But more frequently measurement in the many teams and the many literature you may find, it is the measurement of the total spike or rate diagram. The total number of spikes over all electrodes in every sequence we use 10 millisecond, 20 millisecond or 50 millisecond. It concerns spreading the excitation through the chemical synapses. And as a result, you see on the left side the raster plot of the two bursts, very small bursts, but smallest burst, I think, by the spontaneous activity, as can you see, and evoke activity. Specific size, specific shape of the evoke activity we may measure and choose through the all other spontaneous activity, because as can you see, are very similar. And raster plots of electrical spiking activity we measure through the over 50, 64 electrodes, 60 electrode, maybe 126 electrode, 156 electrode, and so. It depends on the equipment. And total spike rate diagram visualizes the general characteristics of this network, plus give our information about the amount, the blue color, the upper graphs, the amount of burst. Burst may be small, may be super burst, and appearance of the super burst say us about the adult culture, which work like the adult mice. So, if there are no, in this time, for example, microelectrode array, we use optical imaging, you know, we start to use this optical imaging very early. And as can you see, I especially take the same culture and see calcium image and electrical image. As can you see, despite calcium image more really slowly than electrical activity. We measure calcium inputs in the 100 millisecond or second. Raster plot only millisecond. Despite that, we may see if there is a lot of burst, we may measure burst and in the optical calcium imaging. We may the same form the raster plot for the optical calcium imaging. Why sometimes we need to do parallelly electrical activity and optical activity, because here a lot of time resolution, but fluorescence image, it is the space resolution. We may measure any neurons like were in the measurement were in the hippocampus of the mice in the presentation of Olga. On this picture, you see the growth of the growth of the culture during the 21 div. What does mean div? Day in vitro. As can you see, we may measure the first formation spike on the in this network on the about 3 days, 4 days. There are no network. 5 days. As can you see, there is appearance of network burst. Before that, we see only single spikes without formation networks. But here you see really formation of the burst. I think that if we start the beginning from the very dense work with the dense culture, the first burst was appeared on the 5th day. But if we take very rapidly localization of the neurons on the plate in the network, we may see the first burst on the 10th day. But between 10th and 12 days, maximum before the 14th day in vitro, we see transformation electrical synapses in the chemical synapses. And during these days, we see a lot of variability of the size of this network. It's maybe more, maybe less, and constantly changing during the one day. It means there is constantly reorganization of the network for the stable network. Before the 14th day div, there is not only formation chemical synapses, there is traveling of the astroglial cells during the place on the well, and the neurons sit on the astrocytes and, like on the horses, move with the, these astrocytes. And if at the first day there is formations like the ganglion cells, composition of the like in the ganglion cells, body to body. Before the 14th days, we see a random neural network where the neurons sit in some places and be in this stage before the 21 days. As can you see that complexity be rise. If on the left side you see some very simple network burst, here you see very complex network. And if we see the one line, it not means that it is one spike. It's a lot of spikes during the 300 milliseconds and more. As can you see on this slide, you see the change in some parameters like the burst duration, spike burst, burst interval, burst frequency. As can you see from the 5th day before the 15 days, there is some increase in activity. After that, there is quite enough stable period. And there is increasing spike per burst. It is evoked to complexity of the network. And in the normal culture, which we usually use in our experiment. And burst frequency, it means that the general frequency be increased and spike per burst will be increased too. As can you see on this slide, from the development of the culture, which is the same increasing the complexity of the pattern of the calcium events. If, first of all, we saw only single events, the next single events more frequency, the next more frequency, and lastly, we see appearance of burst, 1 burst, 2, 3, and so. And these bursts are coincidence with the electrical activity, but the time also. We investigate the ultra structure of the synapses and parallelly activity of the calcium image and from the electrical activity, and see that appearance of the very complex burst and very long burst like here, evoke to appearance in the culture, like in the viva, spine apparatus, for example. And it means that increasing the complexity of the synaptic connections and so, like in viva. So if we consider this network like the network which work in the homeostasis, really there are no any inputs. There are no any changing of the temperature, pH, the content of the glucose constantly are perfused and so. The neurons spontaneously work maintaining its activity according to this homeostasis. What will be if we change this metabolic homeostasis? For example, do hypoxia, remove glucose, increase concentration of glutamate, or achieve the oxygen-free radicals for the stimulation of oxidation, and so. Good. There are no metabolic homeostasis, and there is changing the network homeostasis. But after the adaptation, through some short period, it's about 3, maximum 4 days, this network, which have another structure, will again demonstrate the new network homeostasis, the same burst. Oh, I think that how we consider this phenomena. Well, first of all, we consider that there is some plasticity. We change the on the level of connections between the neuron, between the properties of the astrocytes, microglia include, and as a result, we see a returning network structure, but on the other level, and maintain the working. I think it's very difficult to kill this culture only if you completely remove oxygen, remove ATP, and so. Anytime will be new network, new network. For example, we remove glucose completely. As a result, we get the transforming the normal state of the network to the middle part, and after the 3 days, we our culture continued to work. The culture which wrote about the 8 months uh, was not in the good conditions. We constantly change or glucose or oxygen or some toxic substances. And this culture constantly returned. And on the but other levels, as can you see, it was very uh, complexity system after the 3 days of the repaying of the glucose. We see the same, not the same. The culture change the structure, functionally change, because a lot of neurons are died, but it not work to the structure of the network. Network continue to work. And for example, more interesting experiment when we take the hyaluronidase and remove extracellular matrix between neurons and other cells. In the cell culture, like in the viva, from the is about 10th day in vitro before the 19th day in vitro, there is releasing a lot of molecules of extracellular matrix, and before the 21 days, we have quite enough development molecule system of extracellular matrix. And if we remove extracellular matrix, what will be? It is a work to remodeling of the network, but not destructive, but remodeling. And after the first hours of decreasing activity, from the 3 days before the 9 days after the removing, we see appearance of superbirds. This superbirds very long, is about 55 seconds, very similar like the seizures. Why? Very interesting, because we observe this phenomena only if we remove this hyaluronidase, hyaluronic acid in the childhood. If we do it in the growth, in the adult culture, the phenomena present, but very less. We repeat in viva in the mouse. We take the mouse, the same line, the same time of postnatal development. It was 17th day, and apply inject in hippocampus a small amount, the same concentration of the hyaluronidase, and through the 3 days, before the 9 days, we get seizures in 70% of the mind. It means that sometimes, and despite I studied this culture a lot of time, sometimes I think it is impossible the same that in viva, because it's a different structure. We check the transcriptome of this culture, and the same hippocampus in the mouse in the same time of postnatal development. It's very interesting that transcriptome in hippocampus will be almost the same, but what they are not? They are not expression of pro-inflammation factors. Immune systems completely is absent. But in this culture, it's very interesting. There are no expression of pro-inflammatory cytokines, proteins, growth factors, and so. Despite that, all other factors which concerned receptors level of synapses and navigation of the axons, dendrites mechanism are present. And repeating the same phenomena in vivo was very, very impressive. So it's very interesting. We may take some investigate some mechanism of this phenomena. For example, we check some blockages of the channels and find that not NMDA dependence reason of these seizures, but IMPA receptors. After that, and the half of the effect depends on the calcium channels, water-dependent calcium channels, and then permits us to investigate why MPA receptors participate in this seizures activity. It was the next experiments when we observe post-translation changing in the MPA receptors. These receptors become permeable for calcium and so. No? And really, if we have multi-electrode array, we may not only measure the activity of these neurons, but we may stimulate these neurons almost simultaneously. And after the stimulation, before and after, we may observe different characteristics of this network. As can you see, there are no change in the burst characteristics. What? Amount of spike, duration of spike, but we see inner structure changing in the inner structure of this network. As can you see, before the stimulation, the main hub was electrode number 19. After the stimulation, number 42. It means that there is change in the inner structure of this network functionally. And today, to continue the topic about the selectivity. Yes, we know about the selectivity. We know that we use hippocampus, and we see what will be if we take the different code for stimulation by the frequency and by the space, and maybe we look for some selective neurons which may answer on the different code, and it really was like this. We get evoke burst activation in the some neurons on the electrode, which may answer only on the pattern of the frequency, different frequency, or only on the location of the stimulation electrode. It means that delay between stimulus and response, some duration, includes the transformation of the signal through the network at a special location, because a stable network. We may determine in the network some neurons which may be selective to space or to type coding. Unfortunately, we cannot ask this culture what you feel and how you understand that it is a different cells, but appearance in the cell culture, these properties of the neuron mean that it is hereditary properties, and hereditary properties will be in the culture like in the liver. And here the slides demonstrate this experiment when we take the S1 electrode and S2 electrode. Different signals to this electrode will evoke to frequency stimulation or space stimulation, and anytime we get other burst network, like the evoked network on this activity. And through the all networks, these properties we observed during the 1 month, maybe more, we don't know, because we stopped this experiment. It's about 1 month we saw. And the next very interesting experiment was may we learn this culture or no? Learning means what? We may stimulate this culture, and we need to get the response. And learning includes the reinforcement, what's very interesting. What we take like the reinforcement for the learning. From the point of homeostasis and from point of stimulation by the other frequency, we understand that increasing metabolic activity is not very good for this homeostatic network. And if we remove this stimulation, if the system repeats learning material, we consider like the reinforcement. It's very artificially we understand, but really not only we, but and other scientists did it and use very low frequency activity, titanic stimulation, and anytime get the response which may be stored in this culture is about 4-6 hours, but not more. After that, after the adaptation, I think the general properties of the all cell culture, I don't know what mechanism of this, but after the adaptation, when we stop stimulate this culture through the 6 hour, I think we see short-term memory. After that, there is recovery of the initial pattern of the activity. If we see on the next time, on the next day, we see increasing activity of this burst by electrical activity in this culture. We use glutamate for the stimulation one day, the next day, after day, and we observed that after the series of application of glutamate, but not toxic dose, activating small dose, it is a work to constantly increasing the level of bioelectrical activity. What do you see on this curve the results of the learning? Sorry. One culture which we curve like the red color, it is a bad experiment. It is a failure of the learning. There are no any learning. But the black color, it is a culture which repeat the signals more through this more shorter time than usually. It means that this culture, like the understand that this signal which achieve the network needed to repeat very fast, more fast than usually, because we stop the stimulation, like the some artificial properties of the learning. Unfortunately, we don't know what the mechanism of this learning, but many, not only in our team, but in other teams observed this phenomenon. So when we use the random connection architecture of the dissociated cells, we understand that in vivo organism, there are no this situation, maybe in some local network in one level, when there is some set of the neurons which are formed some local network. But in organism, there are modular, hierarchic network. When more higher level become more complexity, and complexity. And to build this network, we cannot use random connection network. We need use direct connection between two networks, two local networks. So if we consider the most simplest approach, we may take the local network from hippocampus, from the for example, cortex from the hypothalamus tissue, or maybe from the one region of hippocampus, other region of the hippocampus, and connect each other with the special channels which translate the signals from one network to the other network without feedback connections. But first of all, we do this approach. It's very difficult to grow the next network when this, after the getting information network, second network, target network, will send the impulses to the first exciting network. Now in our team, we do the model when there are 1, 2, 3, 4, 5, maybe 7 cultures, separate local network, which connect to each other with the special channels. But for the random architecture, we may use, why not, for the investigation, the rhythmic activity, even for the learning, simplest mechanism. No, as usually, I think it is medical applications because it's very useful for the studying the neurotropic mechanism of some medical drugs, maybe some medical, but some diseases, why not? Because it's maybe we may grow not from the mouse cells, it's maybe from the iPS cells, differentiated iPS cells. It's very simplest, and we may investigate the molecular mechanism of the synaptic plasticity here, but without very good conclusions, all right, conclusions about the activity of the network, like in the vivo organism. Because even in this picture, you see, it is the fluorescence activity of the calcium image in the hippocampus culture and hippocampus. As can you see, even on this picture, you see the bursting, rhythmically bursting, like in our burst, electrical activity burst. As in the even hippocampus, I don't say about the cortex and any reflex arc from the spine before the cortex. There are hierarchically neural network when one level influence on the other level and only sometimes get the feedback if need. As a result, we use the special approach, microfluid method from the 25th years. There are a lot of microfluidic method, and we did use this method for the formation from the special very transparent materials, the microfluid form, which we apply on the microelectrode array. It's all in 1 millimeter square, as can you understand? It is the needle part, with this part is 1 millimeter square. And we apply some model in 2 models, between them the channels. This picture we take from the literature, from the Brewer, and try to do the copy, and we observed. The neurons axons from the gyrus don't want to pass through this channel before the next work. Don't want, did not want. Why? Because can you see, it is channel cylinder without very smooth walls. And it have done more later in our group that axon cannot pass through this channel before the next, because it's not very useful for the tract geometry of this channel. a lot of time. It's about, I think, 3 years. We try, adaptate, find some useful geometry of this channel, which permits to growth axon from source to target, but not from the target to source. It was very difficult. And I think it's about pictures, I think forms is about 20, maybe 30. We constantly investigate. But lastly, we found that it's very important to grow the axon in one direction if it will be some angle is about 45 degrees. And at last, from target to the source cannot be growth because it is this segment like the cutting these axons because very wide. This very arrow, this very wide. No, as can you see, very useful, very famous phenomena when in the very arrow channel there is increasing the signal, the same signal but increasing amplitude. I think that in the nerve trunk, you know the diameter very low, the same, yes, and the translation signals from one cell to other cells will be significantly increased in the amplitude when travel through the nerve. I think it's very important for the living system. And as a result, we use a special size of the channels which permits us very exactly provide the spread of excitation from one network to other network only in one direction. And what we saw on screen when we check the two network. Вот одна система, это вторая, между ними каналы. То есть видно, что увеличение амплитуды burst, это не значит, что это burst стали какие-то больше, это просто чисто физическое явление, заряд в более узком месте, соответственно, энергию не тратит и сигнал увеличивается. Но смотрите, возникшая пачка в первой культуре через эти каналы передается во вторую культуру, и во второй культуре эта пачка может дать evoked burst network, а может не дать. Условия мы изучали отдельно. Почему иногда она передается, а иногда не передается? Вот здесь на левом графике видно, что при передаче сначала работает... Подойти, я увидела указку. Вот, то есть может, смотрите, вот это вот синее, это то, что вот здесь возникло, красное – это то, что возникло вот здесь в каналах, а зеленое – то, что evoked potential возник здесь вот во второй культуре. То есть мы можем формировать не только evoked potential, как мы привыкли, когда мы смотрим вызванный потенциал, мы здесь можем посмотреть network evoked potential. No, evoke potential cannot be it. We like the evoke potential, but it's evoke network burst. It cannot be potential. Да, because what we measure here, we measure the extracellular action potentials, we understand, in the microvolts. And see, так, у меня нету записи. Вот, sorry. You may see here how the axon try to achieve the second neuron. this neuron need to touch the walls, the next turn to the about some maybe right angle and meet the next arrow walls. Enough will be circulated like here and cannot grow. So we may see on this picture that if we stimulate not only spontaneously propagation, but and stimulation, if you stimulate the cells, we see appearance evolved network in the target. If we stimulate the target, we cannot see anything in this because there are no axon dendritic connections, only in one side. Here we very good visualization through the pattern spiking signature. You see rat plots, how the spread spice, which after the plating on the well was has a lot of big super burst. Some of the culture have no this super burst. So if the culture, despite almost the same, the cells the same, the conditions the same, all standards, but if the cells did not form polysynaptic good network in the source, in the first culture, cannot be transformation information to the second culture, never time. As can you see on this picture? This means that we see in type 1 a lot of larger bursts, very complex forms. The more complexity of the burst, and means the more delays, it means the more synapses present in this burst. This culture may transform through the channels information about this burst to the other culture, and culture repeat this information. But in the weak culture, the first, it's impossible. No, as can you see chemically, I visualize here the stronger polysynaptic connections in the local network may evoke to effective connectivity and transformation the signals to the other local network. As can you understand, between the one local culture and the second local culture, and with these channels, there is one network. You need to understand, big network, which include two local network, like in the organism. There is one level, there is other levels. And for us, will be interesting the next fact, how the second target network constantly getting, but not in the below, but up, constantly getting information will be changing, or maybe not changing, maybe it's only short-term plasticity, and there is no any changing. For improvement of this proposal, we do special chip like Pavel Miloslavovich said us many years ago, do the chip with the different chamber of the medium. One medium must be other medium. So previously chip include two different local network, but in one cultural medium. It's not very advantageous because it cannot permit change the activity of one culture. And if the second culture must be stable. So we do special chip when we may separate the medium of the first culture and medium of the second culture. As a result, but microfluidic technique cannot permit mix this culture or this medium if we apply in the first culture blockages of NMDA and IMPA receptors and switch off the activity, it's a work to change in the activity of the culture, second culture, because their complexity become less. And means that, do you see, it means that, no, for us, it means that the constantly from the birth, from the plating, from the first day in vitro, this first culture stimulate the second culture. And as a result, the second culture must be adapted to constantly activation of this, of its own activity. The second culture, as can be seen here, continue to work with the BIOS, okay, but the structure, functional structure of this work be differ from the previously state when get impulses constantly from the first level, from the first culture. So we count the amount of cells, glial cells, neurons, excited neurons, inhibitory neurons, and found. And plus we counting, we had count the synapses, inhibitory synapses and excitatory synapses, and found that there are no changes in the amount of neurons, the types of the neurons in both culture after the constant action on the next target culture. But there is only one mechanism of the change in functional connections in the target neural network, reduction of the density of inhibitory synapses on dendrites. And it's very interesting if more detail, there are specific spreading of this localization of inhibitory neuron, you know, more near to soma, to axons, the initial segment of the axons, and there is special plus reduction of the density. There is specific spreading of these synapses on the cells. I think as a result, maybe it is a mechanism of the adaptation of the second neural network for the constantly getting information from the other network. No, one of the mechanisms. The next very interesting experiment is only for interesting of us, I think. We are interested in this experiment, I think, have no any task. We test. We take the four chambers and do and think. If we do the circulation network, Lorenta de no, why not? We circuit this through the channels. We circuit these channels, as can you see? We circuit these chambers, four chambers. In all chambers will be the same amount of neurons, glial cells, and so. It's very, very exactly standard. And we stimulate by electrical stimulation the C or this chamber, like here. And observed there are no any circuit will be stopped on the A chamber. Good. We remove some GABAergic synapses by bicoculin. We take different 5 micrometer and 100 micrometer concentration, millimole, micromoles bicoculin, and as a result, we get this circuit. We remove, do more excitable this network. As a result, we get this circuit. But when we increase more inhibitor, we stop circulation. I think maybe due to refractory period, but it's our proposal. We don't know. То есть then more cannot be. We increase, improving, and stop. Maybe previously network was so excited that cannot be stopped after the getting again the signal in this neural network. Now we use free segments, free modular network. We applicate here the neurons which we get from the iPS cells and observe how this apply in this place between two neurons and their connections. First of all, we cut. As the next, we replace the new neurons from the other cells. What will be? It's very interesting. No, it's the model of the repairing of the spinal cord, for example, or maybe in the after the ischemic stroke. Why not? We may use here any cells. No, it is, I think, medical application, but it's very interesting how this new neuron, absolutely new, the days of the growth and of the rising new. Sometimes we use only stem cells, but not stem cells, progenitor cells. How progenitor cells will be applied in the working of this network. Maybe do more worse, maybe more better. No, still now we almost finished this experiment, but I now cannot do quite not clear conclusions, very difficult. So this network with the direct brain-on-chip communication permits to investigate, I think, many properties of plasticity, really network plasticity, because we may stimulate only one network and see how it's evolved to maybe spike time dependence plasticity, maybe Hebs plasticity. How the network will answer on the stimulation of the first network. It's more useful, but we understand that what there are no in our work still now. Day in vitro. Я говорила. Day in vitro. Сокращен. Short. And means that we may in our culture and in the other cultures, I especially saw some literature and cannot find some material about the why. We cannot model, we cannot do the model when we take one network and we get a lot of inputs from the other sides and constantly stimulate from the different inputs, or maybe together, the one of this network. I think that it's very more similar like the inputs in the brain of the animals in vivo, because in our culture, we only maybe 1, maybe 2 times per day stimulate, but not constantly. It's very difficult technically to do, because we need put this culture in the incubator and incubate very most and all electrodes be damaged, unfortunately, but still now. But we show that we'll do this experiment with some modern technique, I think. In this case, we to prove how this sleep network burst be distracted and why be distracted and how the next be summated. A lot of fantasy maybe here. Many applications. A lot of people use this scheme for the therapy, personal therapy of the iPS cells. Use what I think is a very interesting play. The students may take this contact to play in this commercial site with this network. You may open the site, find the useful for you some network, and try to stimulate, depress, and so. It's very interesting. It's commercial game, I think. No, for me, it's game. Memristor, it's more useful information you may get because we may combine the living cells and memristor materials and learn the memristor to answer using not synaptic connections, but resistance, maybe. And so, and conclusion. I think that further development must be linked with the optical, mechanical, electrochemical sensors. It's very fantasy to use some sensors, because in the literature I found recently some paper when this culture network play ping pong. Yeah, I think many of you read this paper. No, still now I think it's only game, because we have no yet other instrument for the maintaining realistic environment of this culture network, because there are no blood-brain barrier. We use diffusion. There are no sensory inputs in the real brain constantly present. And so. As a result, in the future, I think must be a lot of experiments. It's very useful for neurobiological and applied application. Thank you for environment.